otherwise we get in deep shit. So more questions afterwards. So the next talk is by the inimitable Mark Metzstein, who's going to talk about nonsense.
So I too would like to thank the organizers for giving me an opportunity to present our work with you today. And it's about our sort of first steps towards this important both biological and medical problem about rescuing mutants, also known as disease genes, by inhibition of nonsense-mediated mRNA decay. So just to summarize NMD in one slide, if you have a normal RNA, of course, it's translated to make a full-length protein. Make a full-length protein. And then if there's a mutation causing a nonsense mutation that causes an early stop codon, also called a premature termination codon, this would, in principle, be translated to make a truncated protein product. Now, truncated protein products are often very detrimental to the cells. We have dominant negative, dominant active oncogenes and the like. So the cells have actually evolved a mechanism by which premature termination codon containing mRNAs are recognized and rapidly degraded, thus preventing the expression of potentially harmful truncated protein products. This mechanism is very ancient, and I'll show you a little bit. But the funny thing is, we still actually don't really understand how a cell recognizes a premature termination codon from a normal termination codon, but I'm not going to talk about that in this presentation today. So this is what happens when you ask a student to make a slide for you. It turns out that NMD within the cell is actually a double-edged sword. So it has a very well-demonstrated role that I just mentioned in that it targets harmful alleles, which are truncated proteins, and thus alleviates disease or phenotypes. However, because of NMD's function, and this is where we see it mostly as geneticists, in targeting of partially functional alleles can aggravate disease or other phenotypes. And what I mean by that is it's actually not uncommon for a premature termination codon to lead, particularly if it's late in a protein, to lead to a protein which is truncated but is either partially or even completely functional. But the NMD machinery still recognizes this as an abnormal RNA and degrades it, essentially leading to a complete loss of function. So because of this, it's thought that if you could inhibit NMD, you could express this truncated protein, and thus you could ameliorate genetic diseases. And in fact, it's thought that over 20% of all coding mutations, mutations in coding sequence lead to premature termination codons. And so the scope of this kind of therapy would be considered to be extremely extensive. Now, we're at this joint meeting, I really want to give a big shout out to the pioneering studies that understood the NMD machinery, which were originally carried out in yeast and C. elegans in the 80s and early 90s. So the basis of these screens that we used to identify the NMD machinery was essentially exactly the same as what I was just talking about in terms of ameliorating a phenotype by inhibiting NMD, in which they started off with mutant worms or yeast that contained PTC-containing mRNAs. NMD would essentially cause a complete loss of function. And the various groups screened for suppressors of these loss of function alleles, and they resulted in, as I said, truncated but partially functional proteins, which would rescue the various different phenotypes. And from these screens, they identified in yeast 3 genes called UPF1, UPF2, UPF3, and in C. elegans 7 genes called SMUG1 through SMUG7. And then extensive cloning experiments and the like showed that, in fact, UPF1, 2, and 3 were the orthologs of SMUG2, 3, and 4, respectively, indicating that the NMD machinery was actually highly evolutionarily conserved. Now, one thing which is actually important to think about in this actual set of experiments was there is a caveat to screening for suppressors of this nature, which is that the loss of NMD in these organisms, in yeast and C. elegans, are in fact viable. Otherwise, you would not have easily got suppressors which would stabilize these mRNAs. So, from these original screens is essentially where we've got all our information about what the machinery required for NMD, and by cloning homologs, it's found out that NMD is even more ancient than yeast and animals, but goes all the way back to plants. So it's extremely ancient, and every organism contains what's called the core machinery of NMD, which is all eukaryotes contain the core machinery of NMD, which consists of UPF1, 2, and 3, And then, particularly in metazoans, though also in plants, there may be some auxiliary NMD factors called the SMUGs, we call them, which regulate the core machinery. Now, based on getting these homologs in other organisms and genetic experiments we carried out, has shown actually why, for instance, in Drosophila, we never obtained NMD genes as suppressors of mutations in any suppressor screens we did, which is that unlike yeast and unlike C. elegans, NMD function, it turns out to be critical for viability in complex organisms. So in particular, if you knock out the core machinery of NMD, like UPF1, UPF2, in all organisms studied, plants, mice, zebrafish, this leads to lethality. And we have done a number of genetic screens over the years, and we've isolated essentially mutations in all the NMD machinery. And it turns out yet again that the core components are invariably lethal in Drosophila. Okay, so the question which we set out to address some time ago is why is NMD required for viability, right? So there are two actually broad models for this, and they're not really mutually exclusive. So one is that there's every organism contains in its genetic background a bunch of premature termination codon containing transcripts. Maybe they're from inherited mutation, maybe they're from missplicing or other defective from pseudogenes and like. And if these are all got rid of by NMD, and if they accumulate, they block development and viability. There's another model, not mutually exclusive, that NMD there are certain genes in the genome which are encoded to be targeted by NMD, and the negative regulation of these genes by NMD is critical for viability. Thus, if you lose NMD, these genes go up, thus inhibiting development. And so we set out to try to answer this question, and the model we really focused on is this specific targeting model. First, because in some ways it's more interesting to find specific targets, and secondly, genetics on genomic noise is extremely hard to do. So we chose what was likely to be the solvable problem. So what we chose to do was a genetic screen for critical NMD targets, and the way the logic is here, NMD represses a bunch of targets, of which in red are the actual critical targets. It may have other targets, but are not important for viability. And then in NMD mutant, these targets increase, and this somehow blocks viability. So the idea, if you can knock out this target genetically, then you would then rescue the viability of the animal. Now, we did not expect NMD to totally have a single target, and so we actually sort of designed the screen to be much more sensitized than just having to restore viability to NMD mutant. We used a partial loss of function allele of UPF2, which is about 80 to 90% lethal, so we could easily monitor for increased viability. And the other big thing we did is we used the Drosophila collection of molecularly defined deficiencies. And so what we're going to do is actually reduce the targets by half, not by 100%, but this means we could also identify targets that were in themselves vital genes. So we screened about 450 odd deficiencies that covered the autosomes, and it represented about 67% of all bases and 64% of all genes should be covered by this screen. And we identified a number of suppressing deficiencies, and then by ruling out certain intervals because they had overlapping deficiencies and other experiments, we eventually focused on 3 regions. This one turned out to be real. 3 regions that had deficiencies which should significantly suppress NMD lethality. This corresponded to about 60 odd genes. So we had in other experiments defined a set of NMD targets in Drosophila, and so when we took these NMD targets and cross-referenced them against all 60 genes here, we found there was a single direct NMD target Drosophila which mapped to one of these deficiency regions, and it was mapped to this gene called GAD45. So what is GAD45 and why we're excited to find it? So GAD45 stands for growth arrest and DNA damage, and it's quite well known for its transcription to increase under various physiological stresses, radiation, chemicals, virus invasion. And what it does, it activates a MAP kinase pathway through direct interaction with a gene called MEK1, and that leads eventually to a whole kinase cascade and leads to stress response genes, which, for instance, inhibit the cell cycle or lead to apoptosis. And also, one reason this was very exciting is that the mRNA, as I told you, is a direct target in flies, and it's also one of the extremely few conserved direct NMD targets between flies and mammals. And the other thing that was interesting is that cell cycle arrest and apoptosis are characteristics of loss of NMD in flies. So, and if that would be the case, if you lose NMD, this goes up, would activate a stress and apoptotic response. The other thing that was very exciting is the second suppressor region actually contained the MEK1 kinase, the direct downstream target of GAD45. And so to find two genes in the same pathway was obviously very encouraging. So what we went on to do is isolate a null allele of GAD45. This was a couple of years ago, so we did it by the now old-fashioned technique of p-element excision. But we got a deletion which covered the entire GAD45 coding region and no other genes. Now, very importantly, this mutant is homozygous viable, and as homozygous, this is completely viable and fertile, and this is because GAD45 is only required under stress situations. It's not required under normal life situations, but it allowed us to do this experiment, which is now we looked at GAD45 null mutants in an NMD background, and we got this, what was really quite an amazing result to us, which is that I told you that UPF1, UPF2, and actually SMUG5 mutations as homozygotes are 100% lethal. But now, if you make a double mutant with GAD45 homozygous, you actually get a significant amount of survival. In fact, for some, it's all the way up to 80%, 60% for UPF1. And we got essentially identical results with a MEK1 deletion. And also on a cellular basis, I mentioned that loss of NMD, where this UPF2 allele leads to apoptosis, and you see increased cell death here. And this is completely suppressed by removing GAD45, so both an organism on a cellular basis. So what we propose is in Drosophila, in fact, NMD is only required for one function, and that is to keep GAD45 off, and if it's lost, this goes up and it blocks development and viability. Now, also, I'd mentioned that GAD45 is a direct NMD target in mammalian cells also, so we did this experiment in collaboration. We took two different types of mammalian cells, and what you can see is knockdown of UPF1, thus removing NMD, is quite detrimental to mammalian cells. But then if you knock down GAD45 along with it, you increase viability of these cells quite substantially. So it looks as though this is a conserved function of NMD. So in conclusion, we want to propose that you can combine these two regulatory pathways as a method to treat genetic disease. You can either inhibit NMD, which would be otherwise detrimental, but if you block GAD45 that mediates the stress response, you can then rescue these animals. And with that, I'd just like to acknowledge this work was mostly done by John Nelson, a graduate student lab who's recently graduated, with other people, some collaborators, and funding sources. And I chose the most inappropriate picture I could find for Florida, which is the snowy Bryce Canyon in Utah. And thank you for your attention. I'd like to take questions.
Tom.
Guys, that was really nice. But it begs the question,
Speak into the microphone.
Why are the different NMD elements that are essential for NMD, why do they give different degrees of suppression by your GAD, whatever it was?
Oh yeah, so that's a good, so why do different NMD components have different levels of suppression? So one possibility is that some of the NMD genes are actually probably involved in some other processes, maybe translation, for instance. So you're looking at sort of residual, their other functions is our best guess. We don't know that for sure yet, but and I'm not saying that GAD45 is the only target. It's the only critical target. There's probably a buildup of other crap in the genome which is interfering. Yeah, so the question is, does the phenotype of the escapers look different? They do not look obviously different, but they also something I didn't mention is that the suppressed animals are 100% sterile. So NMD is doing something in the soma which requires GAD45, but it has something GAD45 independent in the germline. And we're really interested in pursuing that given all the interesting RNA processes in the germline.
We're only going to be able to take one more question.
Oh. Right here. So very interesting. So I'm wondering, why is GAD45 now a target of NMD? Is there a stop codon somewhere?
No, so there's no real, so why is GAD45 a target of NMD? It's the 3' UTR of GAD45. So it's not an obvious like premature stop codon, but if you couple the GAD45 3' UTR up to GFP, for instance, it becomes NMD sensitive. So what's in that 3' UTR, we don't know yet, but 3' UTR is definitely the source of most NMD sensitivity. So
there's no cryptic upstream open read and frame?
Not as far as we can tell. We've looked at that, and actually GAD45 has no introns actually at all, so it's not an intron-based mechanism. And we've looked, we haven't actually done extensive like race ourselves, but there doesn't seem to be any splice forms of GAD45 at all.
Okay, thanks,
Okay,
Mark.
thanks a lot.